Welcome to Science at FMNH, a podcast and video series that explores the behind-the-scenes science, collections, and research at Chicago's Field Museum. We continue our discussion on emerging pathogens with Shannon Hackett to discover how museum collections play a vital role in understanding biodiversity and its importance.
My name is Shannon Hackett, and I'm the Associate Curator of Birds at the Field Museum. Biodiversity is just a sum of all the life forms that have ever lived on Earth, past and present. I think it's important to remember that biodiversity is not just the organisms that are living currently today, which is a significant number, but it's all of life that's lived on Earth. The collections themselves form the basis from which biodiversity is assessed and/or inventoried. So we need to understand what's out in nature, where it's found, what it is, what it's related to, in order to understand what we might be losing. And the places you go to learn about that, to study that, compared to that, are museum collections. So that's where the library, if you will, of biodiversity is. It's housed in museum collections. In order to know what you have in an environment, in order to preserve it, you need to know what it is. And most people can't know what it is just by looking. They have to do scientific studies. They have to compare what's in nature to the collections that have already been made that represent nature. Scientists sometimes have to take many, many years to actually understand what it is that are in the collections. Frankly, most people are just oblivious and blind to most of biodiversity. I mean, soils, for example, are full of bacteria, but, other than telling your kid don't eat dirt, nobody knows what's in the soils if they haven't been inventoried in that way. So much of biodiversity is hidden to people and actually hasn't even been described to science. When people were collecting specimens 150 years ago, people were looking with an eye to feathers and maybe to conservation as bird populations started declining for particular reasons. It was a tool mostly for identification. The description of new species was really important when people started doing that. And you need to have voucher material in order to do that. It's not good enough for you to just say, well, I saw that and it has a yellow head and something else had a red head. You actually have to document it. And then this process is about documentation. So we went from a period where describing new species was the most important thing, and that's collections were used for, through periods where we got more and more and more advanced data that were taken from the specimens. And then we started taking detailed locality information, and then we started adding weights and looking at gonads inside to figure out reproductive conditions. So we went from that to detailed locality information to making sure we took note of what the soft part colors were. And then technology starts to change so that now we know what DNA is, the last 50 years. So now we understand how important it is to take tissue samples. So we have tissue samples for genetic analyses. And at first those were protein kinds of studies because we didn't have such direct access to genetic material. The actual DNA. And now we take tissue samples with an eye to actually sequencing the genomes of the organisms themselves. And so we're getting basically down to the actual building blocks of life. And now we're saving things with an eye to understanding not just what genes are there, but what genes are being expressed in particular tissues. We are always trying to say with an eye to how we can maximize the things that we want to do with the specimens. And in the case of the Emerging Pathogens Project, what we wanted to do was understand what the biodiversity is on these organisms and how that might change through different kinds of habitats, different kinds of pristine habitats, and then what happens when you go to more and more heavily degraded habitats. What happens to those animals and the biodiversity that lives in them and on them as you change the environment, which is kind of a surrogate for what happens with global climate change. We're just starting to understand from field surveys what that looks like, but we haven't really understood in detail yet from the organisms that live in these environments what that looks like.